Mutation breeding is a planned program for the creation, identification, isolation, and management of genetic variability towards improving cultures. This can be achieved very quickly through this mutation. Hello dear students, I am Dr. Padmavati Rao, Head Department of Botany, J.M. Patel College, Bandar. Today I am going to explain about the mutagens, a mode of action. Contains introduction, inducing mutations, mutagen, mode of action, role of mutagens in plant breeding. Introduction. Generally, mutation is a sudden heritable change occurring in chromosomal DNA, and these mutations cause genetic alterations ranging from single base substitution in DNA to gross changes in chromosomal number and structure. The mutate characters are always true breeding, and these are not found in either of the parents. The characters may be desirable or not desirable. If the characters are desirable, we can introduce in the population. If the characters are not desirable, then we can easily eliminate from the population. So, on the basis of their origin, mutations are classified into two types: spontaneous mutations or natural mutations, and the second one is induced or artificial mutations. The aim of induced mutations. Why we have to induce these mutations? In mutation breeding, we can get new varieties or superior varieties very quickly. That's why we have to induce these mutations artificially. Before going to discuss about this, the mode of action, first we can discuss about the history of these induced mutations. These induced mutations, generally spontaneous mutations, were first identified by Hugo de Vries. These induced mutations were first experiment done by Stadler and Muller in 1927 and 28. They worked on maize and barley plant by using the artificial mutagenic agents like X-rays. In India, in 1957, Dr. M.S. Swaminathan, he worked on mutations at Indian Agriculture Research Institute, New Delhi, and he produced dwarf variety of mutants by using gamma irradiation. In dwarf variety, the yield is very high, and we can harvest this wheat plant within 1.5 months. Generally, in normal wheat plant requires 3 months for its final production or grain production. But in this variety, we can get the grains within 1.5 months. This is the more superior variety. This was first produced by Dr. M.S. Swaminathan and IARI, New Delhi. So, induced mutations, generally two types. We can induce these mutations by using different agents like physical agents as well as chemical agents. The physical mutagenic agents are gamma rays, X-rays, and beta rays. And the most common chemical mutagenic agents are ethyl methane sulfonate, nitrosomethyl uranate, sodium azide, colchicine, etc. Now going to discuss about the mode of mutagenic action, how these mutagenic agents work on the plants or animals, whatever may be. First going to explain about the physical agents that damage the DNA. How these physical agents damage DNA for the creation of new varieties or abnormalities. Ionizing radiation or UV radiation or X-rays, whatever may be, the physical agent generally first works on water, because the water is the main component under any biological organism. If you observe in any cell, 80 to 90% of the cell contains water. So the first exposed agent is water. So whenever the water exposed to ionizing radiation or physical agent, it is split into free radicals like OH and oxygen atoms. These free radicals damage the nitrogen bases and the sugar residues of the DNA. So the DNA, within the DNA, the nitrogen bases and the sugar molecules are present, and these two may break or displace their nitrogen base positions. As a result of that, the damage of DNA takes place. Next, the chemical agents that damage the DNA. Generally, alkylating agents are electrophilic as well as the nucleophilic. In nucleophilic means the nuclear material which has a capacity to absorb these alkylating agents first. Nucleus means it is nothing but the chromosomes. So the chromosomes are first exposed to alkylating agents. Again, these alkalizing agents, they release the free radicals and they are responsible for the breaking of chromosomes at chromosomal level or at even the nitrogen base level. So in some cases, some alkylic agents, they act on hydrogen bonding. You know that GC and AT are formed because of this hydrogen bonding. Whenever these DNA molecule exposed to alkalizing agent, immediately the hydrogen bond breaks down and releases the free radicals. They are responsible for the breaking and disturbing or mispairing of the nitrogen bases. As a result of that, abnormalities occur. So these are the main causes for the damaging of DNA or even the chromosomes. Sometimes spindle fibers are also disturbed. The result of mutagen action. Whenever these chemicals exposed to living organisms, the mutation or the sudden change occurs at different levels. Some mutagenic agents which acts on spindle fibers and causes non-disjunction. Because of these non-disjunctions, numerical aberrations takes place. Means variation in the chromosomal number takes place. Generally, in any living organism, the total number of chromosomes are specific. If any change occurs, the number is varied. As a result of this variation, we will get the abnormality. Like that, if the mutagenic agent which acts on DNA or the chromosome, which causes the chromosomal breakages, leads to the structural aberrations. There is a change in the structure of the chromosome takes place. So these two are the main components responsible for the variation in the living organism. So the mutagen acts on spindle fibers, which causes the non-disjunction. Generally, in normal meiosis, disjunction takes place. Means the chromosomes are simply moving in opposite poles at anaphase stage. This is the normal case. If the spindle fibers are not formed properly, both homologous pairs of chromosomes, they move on the same pole. As a result of that, the gametes having the more deficient and duplicate chromosomes. Means 50% of the gametes having one extra chromosome and the 50% of the chromosomes having less chromosomes. So this gives the abnormalities after fertilization. As a result of that, the aneuploids are produced. The aneuploids means 2n plus 1 trisomics, nullisomics, monosomics, double monosomics, double trisomics, tetrasomics, like that different varieties of aneuploids are produced depending upon its severity for the spindle fibers. So the spindle fibers are very important. If the spindle fiber formation is failed, which leads to the numerical aberrations. Next one is the mutagen which acts on DNA causes chromosome breakage, leads to structural aberrations. You know that structural aberrations means there is a change in the structure of the chromosome. These are of different types, deficiencies, duplications, inversions, and translocations. In every type, the chromosome may break at any place and reunite, or sometimes loss of segments takes place. As a result of that, the chromosome size may change. Sometimes the sequence of genes are also changed because of reunion in the inversions. In all these cases, the mutagenic agent which acts on DNA or at chromosomal level and responsible for the breaking of the chromosome. So as a result, the structural aberrations takes place. So both structural and numerical aberrations are very much useful for the production of new varieties and also the identification of genes in living organisms like plants as well as even in animals also. And we can identify some genetic diseases also by using these mutant varieties. So here the examples of some induced mutant varieties. From 1930 to 2014, more than 3,200 mutagenic plants varieties have been officially released. Among these, 70% of the mutants were directly released and remaining 30% of these mutants are released from their progeny. And according to 2014 survey, 1,000 mutant varieties are already released and they are actively involved in worldwide. Means most of the mutant forms are from horticulture, agriculture, and even in floriculture also we are getting different varieties of mutants, induced mutants. So these mutations are very much useful in agriculture for the production of new varieties. And most of the varieties are very much very familiar type: seedless watermelon, seedless oranges, and flower varieties of gerbera, roses, and dwarf varieties of sunflower. And these are very common type of varieties which are produced by using mutagenic agents. This is the topic of mutagen and its mode of action. So the mode of action is physical agents and chemical agents, how they act on the living organisms for the production of new varieties, means how they act at a molecular level, means gene level or even at chromosomal level. Wherever they act, they produce the new varieties. These new varieties may be useful for the production of desirable plants and even for the identification of genes and mapping of genes and identification of diseases and resistant varieties we can get through this mutation breeding. Thank you very much.